section, we're going to talk about the dengue virus vaccine. So dengue virus is going to be one of these flaviviruses. It's going to be related to the Zika virus and a little bit more distantly related to the yellow fever virus and to hepatitis C virus. Shown here, these are the little virus particles, and shown at the bottom, it causes a hemorrhagic fever, a viral hemorrhagic fever, which can occasionally result in actual hemorrhage from the nose or mouth in particular, in very serious cases. And so this is a serious disease that infects or affects a large part of the world. So if you look at the virus itself, this is the flavivirus that's going to cause dengue fever. And it's going to have a single-stranded positive sense RNA genome, just the one piece. And it's actually going to encode for only one gene. That one gene is going to be then encode for a polyprotein that's going to be chopped up into the smaller pieces that are going to make up the rest of the virion. And 90 of these little copies of the protein are going to decorate the surface. They're going to lay down in little pairs. And then they're going to stand up, and you're going to get 3 of these standing up together, forming a little pyramid, 3-sided pyramid. And they're going to do that to activate fusion. The fusion peptide is actually this little orange thing at the end over here. And so when they stand up, they're going to point all 3 of those little orange things right at the center together. And so it's going to work kind of like a fusion peptide that you know, but these are a different category. They're called a class 2 fusion peptide. And so this E protein is the protein on the outside that's doing all this. And the E protein is the most important characteristic of dengue for vaccine development, because this is the only part that your immune system is going to see. This is the only viral protein that's going to be on the outside of the virion in any substantial amount. So here is the dengue genome. You can see it's non-segmented. It's just one great big polyprotein gene. The structural region is going to be up at the front, and so the E protein here is going to be the one that's on the outside of the virion, and the one that we're most concerned about for vaccination. The rest of the genes are going to do the same jobs as the non-structural proteins of coronavirus do. They're going to copy the viral RNA and diffuse the host immune system. And so other related viruses would be, for example, yellow fever virus. And yellow fever virus, thankfully, has a well-established vaccine. So it is a strain that is attenuated, live attenuated strain, and it doesn't seem to cause disease in people, and it is widely immunogenic. It seems to provoke a good immune response, and it is safe and effective. And frankly, it was just a stroke of luck that people were able to find this thing. So what they did in order to make the dengue vaccine is actually changed out the E gene from the yellow fever virus for the E gene from the dengue virus. And so this is the DEN1, and they're just putting in the 2 halves of this to glue it together and build themselves a full-length virus. So this is going to be a viral chimera, like the mythical creature. It's going to have the E gene from dengue fever virus, and it's going to have all the other parts from yellow fever virus. And the idea is that the E gene ought to be close enough that it'll still work in terms of a vaccine, and you ought to be able to make a nice, strong immune response to this. Okay, so how does dengue work? Well, it's a, positive stranded RNA virus, so let's see. So it's going to go in, it's going to enter via an endosome, something like a clathrin-coated bit. As the pH drops, it's going to escape from that. It's going to release its little RNA into the cytoplasm, which is then going to be translated. It's going to copy itself. We're going to reassemble here at the Golgi, kind of like a coronavirus does, go out toward the surface, and then release the virus without lysing the cell. That's the idea with these viruses. They're very stealthy in how they go about it. And so this chimeric dengue vaccine is actually called Chimera VAX. And it is the most promising current vaccine. It's actually being used pretty widely in the world. And so, the current version, the best version of Chimera VAX is actually tetravalent. So it's a mixture of 4 common serotypes of dengue virus And so this is different than the 4 serotypes we looked at for rotavirus, but there are 4 common serotypes of dengue virus. And it's going to use the yellow fever virus vaccine as its base. So the yellow fever virus vaccine is called the 17D vaccine. That's just the name of the vaccine, a little sort of code name, if you like. And so, yeah, just taking the 17D vaccine, changing out the E gene from yellow fever virus for the E gene from dengue virus, and then growing that thing in, in cell culture, or rather in chicken eggs, is how they actually made this vaccine. And they gave it to monkeys to check if it was fully immunogenic. It seemed to be. They passed the virulent virus challenge, which was really good. And so they said, all right, let's start rolling this thing out. So Sanofi Pasteur were the big producer that acquired the little place that made this. And they were the ones that were going to roll this thing out. Now, the first thing to note is that the virus, which was vaccinating against 4 different strains, actually gave pretty good protection against 2 of the strains, around 75%. Gave about 50-50 protection against 1 of the strains, and a little below 50% protection against strain 2. So it looks like this strain 2, or serotype 2, they needed to find a better way to vaccinate against. But still, we're getting pretty good protection against all the other ones. Now, the problem with this vaccine, it's turned out, is that this is a vaccine that has to be given at a specific time. So with dengue fever virus, you have antibody-mediated enhancement. This is the same thing we saw with feline coronavirus, where if the body makes antibodies, but they're not neutralizing antibodies and they just stick, then you can end up sticking the virus to the cells it's trying to infect, and you can actually get more infection that way and more severe disease. And so if a person just gets this Chimera VAX and they've never had dengue before, what they'll build up will be a range of those non-neutralizing antibodies for the most part. It's random chance, but that's what most people tend to make. And then if they do get the dengue virus after that, it's going to be much more severe, because dengue is a virus where the first time you get it, it's generally mild. But the second time you get it, because you have those antibodies in your bloodstream already from the first time left over, the second time you get it, it will be more severe. And so the idea is that the vaccine is used not at first on what we call naive people who haven't seen the virus before, but it's only used on people who have had at least 1 confirmed case of dengue virus, and then you give them the vaccine. And when they've had 1 full case and a vaccine, that then puts them over the top. That is then enough immunity, because dengue is a virus that you're only going to get once or twice, or not at all. And once you've had it twice, if you don't die from it, and it only has something like a 1% case fatality rate, then you will be immune for life. And so the vaccine is moving people from some immunity, but perhaps dangerously low, into enough immunity to be able to resist the virus for life. And so that's kind of a neat use of a vaccine, I think. Okay, and then our last one, let's just add that onto this section, is going to be the human papillomavirus vaccine, and it is an anti-cancer vaccine. And all the people that have been saying, why doesn't science come up with a cure for cancer? This is literally a cure for cancer. And it's a cure for cancer that you can get paid for by the government in a lot of countries, and even some places in the US. And this is a vaccine that is still somewhat controversial. So yeah, science has cured at least 1 kind of cancer, or been able to prevent it. And boy, yeah, people are complicated. So let's look at the actual vaccine. So this was one of the posters. Human papillomavirus does cause cervical cancer. It's a virus that men and women can get, and it can cause other kinds of cancer. It can cause anal cancer, for example. It can cause throat cancer. It can cause cervical cancer. It can cause penile cancer. It can cause cancer of whatever bits you happen to enjoy rubbing onto somebody else's bits, I would say. It would be a nice way to put it. So yeah, what is this thing? So this is a virus that I believe we've already covered. So it's a little tiny virion, only 55 nanometers, icosahedral shape, double-stranded DNA virus, no envelope, just a protein shell on the outside. There are going to be papillomaviruses that are species-specific, site-specific, and specific to a particular part of the epithelial layer. Like I was saying, they're going to like basal cells in particular. And these two strains, strain 18, HPV 18, and strain HPV 16, are both highly associated with cervical cancer. So these are things that in general are only going to cause genital warts or may not even be detectable on a male. But when they get into the female, they grow really well on the cervix. And so they are associated with a lot of cervical cancer. And so that's what you'd be vaccinating against. And I think some of the pushback against the vaccine is that, if you never have sex, then you don't need to worry about this vaccine. But from my understanding of the world, a lot of people do have sex occasionally, and sometimes with more than one person. And you don't always check full vaccination records. And, honestly, these things aren't necessarily planned out months and years in advance in every case, would be my observation. And so this is why it's good to actually vaccinate everybody. And so the L1 protein, which is going to be one of those late genes, is going to make the major capsid protein. And you actually grow up the L1 protein and make little shells of it. It will assemble itself into a little icosahedral ball, and you can inject those into people. And it's like their immune system is seeing the virus. They make a somewhat weakened but still strong enough immune response. And then they'll have antibodies in the future that will protect against infection with the actual virus. So why doesn't our immune system just respond to HPV infection adequately? Well, HPV doesn't really cause cell death. HPV causes cells to divide. It's a cancer-causing virus. A lot of those toll-like receptors are looking for things that would come out of a cell that's recently died or exploded. Those are general signs that something is not right, and there's danger around, and so we better put up our defenses. So for most of the HPV infectious cycle, there's little or no way that the cell can actually recognize them, and so there are no pro-inflammatory cytokines released. And so there's no way to present this to the immune system, and so it just goes undetected because it's inside of a regular cell that appears to be doing its job. And it's not until that cell is all the way on the outside, about ready to come loose and fall off, that's when the virus actually comes out. And that's a time at which the immune system essentially can't see it. And so that's why we don't do a good job of preventing this. So here is the papillomavirus. It's got to get into this basement epithelium cell. And if you have some friction on the upper epithelial layers, as in sexual contact, for example, then you can get the virus and it can find its way down to the bottom cells where it can infect. And then once it's infected, it's going to cause this cell to differentiate a little more than the others. And so these differentiated cells are actually going to pile up, and they're going to make something like a little wart. And that's what a wart is. It's a little tiny cancer. And each one of these cells is going to have the DNA, and it's going to start making viruses when it gets toward the outside. And then when they're just about ready to fall off and or burst, and they're going to be full of viruses, the viruses come out, and then they can potentially go into somebody else. And so the vaccine looks like this. This is what L1 looks like, and this is the perfect little capsid of human papillomavirus. Here's a whole bunch of them all lined up on an electron microscope grid. Here are some virus-like particles that have been made with just individual proteins that have glued themselves together. You can see they're pretty good. They're not as good as the actual virus. That gets things done, and it's really tight and smooth and regular. You've got a little bit of irregularity here, but it's actually not enough that it causes a problem. These little virus-like particles are, they don't have anything inside. You can see they're all hollow. And so they don't have any DNA. They can't actually cause an infection. This is just an injection with a little bit of protein and a little bit of purified protein at that. And so that's about as harmless as you can possibly get. And it still generates a pretty good immune response and seems to be very safe, very well tolerated, and quite immunogenic. So two of the most prominent ones are going to be the formulation called Gardasil and another one called Cervarix. And Gardasil is going to go for immunity to 4 different really common types. And so it's got 4 different types of those L1 proteins in there. And Cervarix is going after the 2 major causes of cervical cancer only. They're just manufactured by different companies. This is a Merck one, and this is a GSK, GlaxoSmithKline one. And it's going to be made in yeast versus insect cells, which are going to glycosylate a little bit differently, but both of them are just different ways of producing a large amount of protein. So they're going to be very comparable in terms of what they do. I think given the choice, you probably want to go for the Gardasil. So you get more immunity. But then, of course, there were a couple of stories like this. And so you'll hear about people that have died after this cervical cancer vaccination. And these are people whose bodies just made a massive and inappropriate immune response to plain random protein. And this is an extremely rare condition, but it is something that does occasionally happen. There have been a couple documented cases. But typically, when this does happen, the reason why it happens, and this is what they found out later, is that it's because of pre-existing conditions that they just didn't know about. So in this case, this particular girl that died in 2009 turned out to have already had a big tumor in her chest. And so they didn't realize this, and when the vaccine went in, it caused just enough trauma inside of her body that, this big tumor in her chest, something went wrong with that, and that actually caused her death. So that was very sad. But it wasn't really the vaccine. It was really, the combination of any stress, really, and what was already going on there. So how does this work? Well, the human papillomavirus L1 protein, you make this virus-like particle by just letting them associate together and form up little not quite empty viruses. Or they're empty, they're just not quite perfect viruses. And then those are going to be picked up by antigen-presenting cells, things like a macrophage or a dendritic cell in particular. And the dendritic cell will present it to a T cell, which could then, show it to a B cell and get that B cell going, which could make those different antibodies, those immunoglobulins. That's what the IG stands for. And then if it gets good at making immunoglobulins, it will switch classes of immunoglobulins. So there are 5 main types and a couple subtypes that it actually goes through in a particular order. And once it starts producing these antibodies, it'll be called a plasma cell. It'll change a little bit. And then there are the antibodies. And once you have these antibodies, they will block future human papillomavirus infection. This is the idea. And so in vaccine trials, you've got 99.7% of people that get the vaccine actually get a real strong IgA response to this. So this is exactly what you want. And compare that to people who are actually exposed to human papillomavirus. Very few of those, something like 5%, would actually make a strong immune response. So this vaccination is much better in this case than actually having a live virus because of the way the virus grows and the way that it does not trigger immunity. When they tested this out, this is the number of people that got the control vaccine that didn't get vaccinated, essentially, that came down with human papillomavirus. Here's the number that actually came down with it that were vaccinated. And so you can see that the vaccination was actually an absolute slam dunk. This was, really good. And so that's human papillomavirus vaccine. It is available at your doctor near you. And you can go and get this. And it works. It's probably a good thing to do. It's going to be most protective when given to people before they are sexually active. But again, since you never know necessarily. You haven't necessarily planned out, when you're going to start becoming sexually active, down to the minute, years in advance. It's probably a good idea to vaccinate a little bit before that. And so they tend to go for vaccination in people who are about, I think, 11 or 12, maybe 13 years old tops. Like before they would have had a chance to be infected with human papillomavirus from human contact with somebody else. So there we go. That was the human papillomavirus and dengue virus parts of the modern vaccines section.